Hello. Now, welcome to the next part of my talk of the nanopore sequencing. And this next part is the part which deals with the basic principles of the nanopore sequencing. So, what is the basic principle of the nanopore sequencing? Okay. So, we would be talking about that. And then we will be moving further deep down into the nanopore sequencing. Okay. So, coming back. So, बेटा जी जो बेसिक प्रिंसिपल है नैनोपोर सीक्वेंसिंग का वो ये है कि पहले भी बताया दैट सीक्वेंसिंग इज टेकिंग प्लेस थ्रू अ वेरी टाइनी पोर्स. अब इफ वट एवर आई एम हाईलाइटिंग ओवर हियर कैन प्लीज पे अटेंशन ओवर देयर दैट दिस इज अ blue colored with blue borders and white color. This is a membrane, and this is that I'm pointing it out, and I'm writing M over here. This is a membrane, and on this membrane there are tiny holes, and these tiny holes, they are regarded as this one, it is regarded as the nanopore. Okay, if we magnify this whole area, then it will be like this. Okay, so you see that this is the membrane right here, and this is the tiny pore which is present in this membrane. So this is the tiny pore, and this tiny pore is called as the nanopore. Okay, the important characteristic feature here is that कि ये जो membrane है, the membrane that is there, that means the membrane on which the nanopore is there, you have to remember that it is electrically resistant. Electrically resistant का मतलब है कि the electricity is not able to pass through the membrane. It can only pass through one part, and that only one part is the nanopore. Okay, so nanopore में से the electric current can pass, the electrolytes can pass through this space only. बाकी किसी भी part से कुछ pass नहीं हो सकता. Okay, and like that of electric current, the DNA nucleotide, which can either be DNA or it could be RNA, it can only pass through the nanopore. There is no other space where it can go. And remember, you know that DNA is negatively charged. Therefore, DNA, it travels from a region of negative charge and it moves towards an electrode of positive charge. So it moves from electrode which is labeled as negative. So electric field लगाई जाती है. Above this membrane, you can see that there is an electric field. This electric field has two electrodes. One is the negative electrode and another one is the positive electrode. And between this lies the membrane bearing the nanopore. So this is the basic structure of the nanopore. And in that principle, basic structure of the nanopore membrane. Okay, okay, going forward. So what happens next is that whenever this strand of DNA it passes through this nanopore, तो जब भी ये strand इस nanopore के through pass करता है, there is a change in the electric current. Electric current सिर्फ यहीं पे flow कर रहा है. किसमें flow कर रहा है? In the nanopore. But whenever आप imagine करिए कि that ये एक hole है, and this is a hole through which what is passing through this hole, water is flowing from a region of higher potential to a region of low potential. अब imagine करिए कि इस waterfall में आप ball डाल देते हैं, तो जब वो ball pass होती है through that waterfall, through that hole, so a change is produced in that waterfall. That change is monitored, can be monitored. So similar thing is happening over there कि इस nanopore के अंदर क्या हो रहा है, the number of electrolytes are passing through this pore, और जितने electrolytes हैं they are passing because there is a potential difference across this membrane. That is why, and that potential difference can be recorded. तो जब भी कोई extra molecule है, be it be a nucleotide molecule or be it be any molecule, if it whenever it makes change in this flow of electric current, मान लीजिए normal flow ऐसा है, ठीक है, a straight line, normal flow is a straight line. So whenever there is a current, whenever there is a fluctuation in this straight line, it could be like this, it could be like that. Okay, and this change in the electric current can be either be same or it can be different. And this picture over here gives you an idea कि इस तरह से ये जैसे आप ECG करते हैं और उसमें QRS waves होती हैं, and that can be recorded by the electrocardiogram. Similarly, the change in the electric current produced by the ACTG is recorded by the electrocardiogram. ठीक है। सो इवन ACTG अगर RNA होगा तो U भी पास कर सकता है इसमें, ठीक है। अब इम्पोर्टेंट फीचर क्या है, क्या ACTG और U सेम चेंज करंट प्रोड्यूस करते हैं? आंसर इज नो. So the change in the magnitude of the current which is produced by these nucleotides, it is different because the composition of the nucleotides is different. ACTG, they are ampurines and pyrimidine, two categories. Further, they have different compositions. And research has shown that different types of nucleotides, they produce different fluctuations in the current, and therefore if A produces a characteristic change in the current, similarly T produces characteristic change in the current, and so on. Okay. So now, however, there is a prerequisite. ठीक है, अब क्या होता है कि नॉर्मल जो ACTG हैं, the normal nucleotides हैं ACTGU, these while they pass through the nanopore, they do produce change in the current, but the problem is that the magnitude of the current which is produced by them, change in the magnitude of the current which is produced by them must be detectable. मतलब कुछ भी आप चेंज करते हैं वो ऐसा होना चाहिए कि वो इतने वॉल्यूम का होना चाहिए, वो इतने मैग्निफिकेशन का होना चाहिए that it can be detected by the detector. ठीक है। सो इसलिए जब नैनोपोर सीक्वेंसिंग करते हैं, वन ऑफ द प्री रिक्विजिट इज दैट कि हम थोड़ा सा इन बेसिस को मॉडिफाई करते हैं, ठीक है। और वी कैन से दैट we do some modification in the DNA nucleotide sequences. Why do we do that? We do that so that whatever signal is produced by them, that can be detected very effectively. ठीक है। अब इसके अलावा मॉडिफिकेशन नॉट ओनली हेल्प्स इन प्रोड्यूसिंग अ डिटेक्टेबल सिग्नल, इट आल्सो प्रोड्यूसेस cause it does two main other activities. And what are those two other activities? The other activities that it does is that it also brings the DNA fragment in reach of the nanopore. ठीक है। and it also helps in controlling the passage of the fragment through the nanopore. इस पॉइंट को मैं थोड़ी देर में फिर से आपको एक्सप्लेन करूंगी कि why does that happen और ऐसा क्यों होता है. So I'll come back to these 2 points later कि इनकी डिटेल क्या है. Okay, so now what are the modification? Now the question arises कि modification क्या है. So the modification is called as ligation of some adapters which are called as Y adapters. ठीक है, हम कैसे modify करते हैं? The point is, अभी मैं क्या बता रही हूँ कि ACTG, whenever they pass through the nanopore, they produce a change in the current, and that change in the current is detected by the detector. However, the amount of change in the current which can be detected must be of a measurable amount. So therefore, the normal bases hai, they need certain modifications, and certain modifications are also done to achieve two other goals which are there in this slide. However, I will discuss about them later. But modification क्या है, क्या modification करते हैं? जैसे मैंने बताया है कि modification is that कि हम लोग इन न्यूक्लियोटाइड्स पे या DNA sequences पे adapters लगाते हैं. These are called as Y adapters. Now, question is, when does these adapters are added? Now the answer is adapters are added to the DNA sequence during the library preparation method. Okay, let's stop here. Remember Illumina में what we have, and I have talked about Illumina. I have talked that the sequencing in case of Illumina, it has three main steps, and the steps are: first, the library preparation; second is the cluster generation process by bridge amplification, and the third part is sequencing itself. Ab, in case of nanopore, as I have already told you again and again, that the first step is like that of second generation sequencing, library preparation, but second step, cluster generation में क्या होता है? Cluster generation में PCR amplification है, clonal amplification है. Here I have already told about is कि nanopore में we do not need the process of amplification. So therefore, second step is not there. Only two steps are there. First is library preparation and second is sequencing. Okay, and the modifications that I have talked about is introduced during the process of library preparation. So this slide here gives an overview of ठीक है, ये बेटा आपको क्या बता रहा है, दिस इज गिविंग यू एन ओवरव्यू ऑफ कि किस तरह से मॉडिफिकेशन हो रही है, किस तरह से लाइब्रेरी प्रिपरेशन हो रही है. So library preparation, again, as we have discussed in case of Illumina, can be done in two ways. It can be done by the process of fragmentation. If you have a high molecular DNA, then that high molecular DNA को आप क्या कर सकते हैं, you can fragment it, okay, by sharing, or you can also fragment the DNA using the process or help of transposon. तो अगर ट्रांसपोजोन से करते हैं तो टेगमेंटेशन होता है, अगर दूसरे से करते हैं तो फ्रेगमेंटेशन होता है, ये मैंने आपको पहले भी बताया है. अब in case of nanopore sequencing, you may or may not, that is your choice. अगर if you have got a DNA sample which is from a eukaryote और उसको आपने सीक्वेंस करना है, then at that time you might require and उसका जीनोम का मॉलिक्यूलर वेट बहुत ज्यादा है, the molecular genome size is on a very higher side. At that time you can decide to fragment the DNA into small pieces and carry out sequencing of those pieces. However, if you have a comparatively smaller genome, as in case of bacteria, you can just go forward and straight away sequence the DNA molecule. So in here, what you do is, in case of fragmentation process, आप DNA को लेते हैं. So whether it is a fragmented DNA or whether it is not a fragmented DNA, what you do is you end repair it. Keeping in mind कि हो सकता है कि the ends of the DNA are not blunt and they are not clean, they are not sharp. So in order to modify the DNAs, you do a process which is called as end repair, highlighted over here. So what do you do? End repair. End repair में you make the ends of the DNA blunt. बिल्कुल सीधा बना देते हैं, staggered नहीं रहते. You make the DNA blunt, and once you have made the DNA blunt, then you add the enzymes. They add a poly A tail to the DNAs, as you can see over here. Okay, these poly A tails are deliberately added. Why they are deliberately added, or A overhangs are deliberately added, so that you are now using the A overhangs, you are able to add the sequencing adapters to this DNA fragments. So this is the sequencing adapters that is being added over here, and this is the 3'. Okay, once these adapters are called as Y adapters, as you can see that इनकी shape जो है वो थोड़ी बहुत Y shape होती है, so therefore they are called as Y adapters. However, in case of tegmentation, wherein the shearing is done with the help of a transposon, what is done is that there is a complex called as a transposon complex. This transposon cuts the DNA into small pieces, and in that process, like that of Illumina sequencing, it adds the adapter called as transposase adapters. Once these transposase adapters are added, then they are used as a template to do a PCR wherein the sequencing adapters are added to the DNA molecule. And if you see clearly, then again Y adapters are added to the 3' and the 5', and like that of the fragmentation process or simple fragmentation without the help of transposon. ठीक है? Okay. So this next slide. So this is how the DNA molecule after the library preps looks like. This is the what they have done is that actually they have added the adapters. We have added Y adapters on either side of the DNA. So this is the Y adapters, and you can see there is a yellow blob, there is a black something over there, and there are some blue stretches over here, and we will talk about it. Okay, so library preparation में deliberately we are adding the sequencing adapters or the Y adapters. Okay, what happens next? Before I talk about it, again, this is an enlarged view of the fragmentation process. You can see here that जो DNA था